Okay, everybody. So, how are you guys doing today? Can you hear me? Yep. It is Friday. Yay. Okay. So, um, I did get the eye clicker to work. All right, guys. Let's quiet down. Let's get into our cell bio zone. Right into the Zen zone. You are a very chatty crowd. I will give you plenty of audience participation. All right. All right. So quiet. It's my mean voice. All right. So I try. Okay. So here, uh, some of you asked me before about the dates for exam number 4 and the final exam, which remember the final cumulative exam is optional, right? Remember that if you have good grades from 4 exams, you don't have to take the cumulative final. If you have one bad grade and you still pass in the class, you can get away with not taking the cumulative final, which is totally fine. It depends on how high you want your grade, right? If you bombed one exam and you want to make it up, plan to take the cumulative final, okay? And I'll tell you something else. Are you listening? Yeah, the questions on the cumulative final, I picked them from the previous exams. So they are basically the same questions. A little bit of modification here and there, a little bit, not too much. Okay? So if you understand the subject and if you, review your exam and you really are on top of things, I would tell you, take the cumulative final. It's not going to hurt you. Okay? Taking the cumulative final, you will drop one lowest grade, one lowest exam grade. Everybody cool with that? All right. Day 4, exam number 4, and the cumulative final, I have added on the syllabus. It's going to be on December 10 in this room from 1:15 to 3:45. This is giving me such an echo. All right. So 1:15 to 3:45, how is it that works? You're going to come to class, bring 2 blue scantrons. For the first scantron, well, fill up your scantrons before coming to class so you don't waste time cleaning those bubbles, okay? For the first scantron, you're going to do exam number 4. So we'll give you exam number 4. You finish exam number 4. You hand it in, show ID of some form. We have to make sure you are who you are, taking the exam. Hand in the exam, and then you tell us if you want to take the cumulative final or not. Okay, if you do, then we'll hand you this number 5. You go back to your seat, take exam number 5 using the other scantron. Then you hand it in. And believe me, this is plenty of time for that. Okay, so everybody clear with that? All right. Okay, so just to wrap up on the proteins that I was rushing a bit through those last slides. So just to make sure that we have a good idea on protein function and control of protein function. I had mentioned before here, control of GTPase activity, right? Remember you have, well, as you're gonna see later, for signal transduction. Guys, I don't like to talk over chatty crowd. We'll put in my main voice. Okay. So, because I have a very short attention span and everything kind of distracts my head. So let's be quiet. So I'm going to get you some very cool information today. All right. Control of GTPase activity. Remember I told you about enzymes? If it ends with A's, it's most likely the name of an enzyme, right? And the name of the enzyme is going to tell you what it's doing. So this thing is saying GTPase. What do you think it's doing? Breaking down GTP. Okay, so how do you control GTPase activity? Control the enzyme that is breaking down the GTP. You're going to have regulatory proteins that are going to work on either keeping GTP there or putting GTP there or degrading GTP. Okay? So here, you have this enzyme that is called GTPase activating protein. Okay? So this is a protein that is going to push GTPase to do its job. Okay? The short name for GTPase activating protein is GAP. Now, when your protein is bound to GTP, the other protein, okay, so this guy is GTPase GAP, it is going to act on this other protein here. So this is another protein, right, that is bound to GTP right there, okay? When this G-coupled protein is bound to GTP, this blue stuff is active. When GTP breaks down to GDP, then this blue stuff becomes inactive. Okay, so breaking a phosphate out of the GTP that is bound to this guy here is going to deactivate this GTP coupled protein. Okay, so this guy here, GAP, is going to go in here. So let's give it another color. Let's put it a green color. GAP is going to go in there and is going to push the GTPase activity of this guy right there. Okay? So when GAP goes in there, it pushes this thing to break down GTP. So the thing that's going to be bound here now is going to be GDP. So GAP is actually turning off the activity of the GTP coupled protein. Okay? So this is what it's saying. All right. And then there is another protein that is called guanine nucleotide exchange factor. So this guy is called GAF. Okay? So what this guy does, it does the following. Okay? So here you have your GTP coupled protein, okay, that is now bound to GDP right there. So this thing here now is GDP. Why? Well, because it got hydrolyzed from GTP to GDP. Okay. So now let's say that you need this coupled protein, this guy here, you need it to be active again. So then comes this guy here, guanine nucleotide exchange factor, okay? And this guy goes in there and it's going to exchange GDP for GTP. So it's going to take out GDP and it's going to put back in there the GTP. So this is all it's doing, is exchanging GDP for GDP. Okay? So that's your GAF, is your exchange factor. Then when this coupled protein is bound again to GTP, then it becomes active again. Right? So this protein here is going to downregulate the action of your G coupled protein, and this guy over here is going to exchange GTP for GDP on coupled protein, and it's going to make it active. Okay, so this goes down, this goes up. How we're going to differentiate which one is making the coupled protein go down, which one is going to go up, you know. Okay, so this is explaining what it is. Said this. So I put this picture over there. Anybody been to London? I mean, to the tube. That's what they call the subway, right? In England. No. Nobody? Oh, one person. Okay, two people. Well, I've been there once. Yeah, three people. Okay. I've been there once, and before you get into the subway, the tube, you know, there is this lady who talks about mind the gap, mind the gap. You know what I mean? Yeah. So I put this picture of the guy that did not mind the gap and kind of got deactivated very close to the gap. Maybe that will help you to remember gap is deactivating the GTP coupled protein. You know, that's one strategy of remembering things. You associate it with things that are either funny or ridiculous or both. All right, whatever helps. Okay. So here, the last part on protein Regulation was this part on adding stuff to the protein or having an environment that the 3D structures don't fit very well. Okay, so I put those two buckets into one bucket here. So you can add groups to proteins that are going to tag it to do something. Okay, so you can modify your protein to tag to do something. In this case, I'm putting the example of ubiquitin, that is this little protein over here. Okay, it happens to be positively charged. Okay. So in this case here, ubiquitin can be added to a lysine residue on a target protein. Okay. So it's not at the end of the protein, it's not at the beginning, not at the carboxyl or amino terminus. It's in the middle residue somewhere if it has a lysine. So you have a possibility of adding this bulky ubiquitin tag to this protein. Now, adding the tag can do one of several things. Like, for example, if you have a protein that is involved with regulation of histones, as we're going to see, histones are interacting with DNA. Okay, so if you add this to a histone, you can regulate the activity of a histone. So usually you put one ubiquitin and you're going to regulate the kind of histone activity. If you put several ubiquitins in different sites of this target protein, you can trigger the cell to undergo some type of endocytosis. Okay, so it's going to start eating this stuff. Okay, and on the other hand, if you put several ubiquitins like tagged, like in a little chainsaw here, okay, then you actually tag the protein for degradation. That's usually what we hear about ubiquitination. It's like protein degradation. The more ubiquitin it has tagged, the faster the protein is going to get degraded. Okay. On the other hand, you can also have other proteins that can get this little, train tag of ubiquitin, and the protein can be triggered to go and do some repair in the DNA inside of the protein, inside of the cell, inside of the nucleus. So there are several uses for ubiquitination. So it can be used for signal transduction, for protein degradation, cell cycle regulation, endocytosis, or autophagy. You know what autophagy is? Is when when the cell starts to eat its own contents, to recycle its own contents. Yeah, so the cell kind of like eats itself, its contents to recycle its stuff, and it's actually very good for cell turnover. So the older, more deficient cells are the ones to get autophagy, first. Okay.
What's that?
Ubiquitination. Yeah, it's a big word, and everything is in your PowerPoint. Oh, by the way, I just have opened all our previous PDFs on the modules, and I also open all the links to my to last year's live classes. Okay, so if you want to watch the lectures ahead of time and get more, acquainted with what's going to be taught in class, you're welcome to go and follow that secret path to watch the other lectures. Okay, but remember that every year I modify a little bit the lectures, so it's not like you can just follow whatever is there and then you're going to be totally 100% fine. I might focus on different things in the class, so you still have to show up. All right?
Yes.
Okay. Yeah. Oh yeah. I'll put this year's on top of last year's. So it depends on how much YouTube can stand. Yeah. Yes, but my last year lectures, they are on 2024 playlist. So this year is on 2025 playlist. Yeah. Oh. Actually, last year's lectures, they are not public because the laws changed this year. So I have to, now they say they will make everything public. So, okay, so no secrets, so everything is there. Okay. Okay, so here is a slide just summarizing the protein modifications. Okay, so it's just everything in one slide. So you have ligand binding, that's your allosteric factor. Remember, allosteric factors can be your cofactors or coenzymes, right? You have phosphorylation. Remember those 3 amino acids that can be phosphorylated? Remember what those are? They have to have hydroxylic group on the side chain. There are only 3 of them. 3 of them you have to remember. What are they? Serine, threonine, and tyrosine. Okay, so you got to remember those have hydroxylic groups, and those are the ones that can be receiving that bulky phosphate group. Okay, and the reason that you need the hydroxylic group in here is that you are going to take out that hydrogen and you are going to add your phosphate that has those oxygens, right? The big electron cloud of oxygens over here. So in order for this thing to work, you need this thing to be formed. That's why you need the hydroxyl group over there. Okay? And those 3 amino acids are the ones that can do that. So the other type of control, GTP to GTP switching, and then the last one I just mentioned is mechanical or covalent modification. As I promised you, I did get this iClicker to work, so you're going to get a lot of extra points today. Yay! So let's get your iClickers ready. Okay? Yeah? AA. It's always on AA. As far as I can remember. Okay. All right, so first question. During an enzymatic reaction, the substrate binds to which of the following? So you gotta remember where is it the substrate is binding. Option A, the site near the hydrophilic region. Option B, the active site of the enzyme. Option C, the site near the hydrophobic region. Option D, the location rich in sulfur-containing amino acids. Okay, so let's open this. Okay, so this is the time when you can actually cheat. You can talk to your classmates. Make sure you get the correct answer. Okay, 3, 2, 1. Oh, it doesn't stop. Does anybody need more time? No. Okay, what's your answer? B. That's your active site. Okay. Another question. How? In a reaction rate versus substrate concentration graph. So here is reaction rate, that's your velocity, versus substrate on the x-axis. The substrate concentration at half of Vmax is represented by what? Okay, does anybody need more time? No? What is your answer? C. That is your Km. Okay? The concentration of substrate at half of Vmax is going to give you Km. And what is Km a measure of? Enzyme affinity for the substrate. Km is a measure of the enzyme affinity for the substrate. If it grabs the substrate at very low substrate concentration, so your Km is going to be small. If it needs a lot of substrate concentration, then your Km is going to be large. The larger the Km, the lower the affinity for the substrate. Okay, so now I'm going to try something interesting. Confidence. All right, so those of you who are using the app on the cell phone, okay, I'm going to press this confidence thing because I want to tell you when you answer this question, how confident were you at giving me the correct answer? Okay, so I just put this confidence on. Did it change anything on your app? No. Oh, maybe I have to put it before the question. All right, let's try for the next one. Got more questions. Here we go. Next one. Okay, so now the confidence thing is on. Let's see what happens now. Protein phosphatases do what? Okay, so you should put in your answer and tell me how confident you are. And this is an easy question, right? You have basically 50% chance of getting it correct. Does anybody need more time? No? Okay, so I'm going to stop it. What is your answer? B, it will remove phosphate. Okay. Do I have more questions? Okay, not now. All right, so now finally we're going to get into chromosome structure and genomes. And finally, here to chapter 4. So you are, in case you haven't seen the DNA structure before, you're going to look at it again, okay? And then you're going to look at chromosome compaction, how it gets folded. And then we're going to look at chromatin and DNA function. Okay, so when you look at a cell in the microscope and you focus in the nucleus, you usually see chromosomes. There is a mush of stuff, and you can't quite tell which chromosome is where, okay? It's only when you start cell division that you get chromosome condensation and you can actually tell them apart. So this cell here is undergoing, the cell division is undergoing mitosis. So in this case, you can actually pinpoint where your individual chromosomes are because they are condensed and they're being pulled apart. So in the non-dividing cell, the whole thing is just a mush. Unless you stain the chromosomes, you can't really say which one is which. Okay. And the question here was, how was it discovered that DNA was really the genetic material, was the information for heredity? Okay. So there are a couple of experiments that are those classical experiments that you should absolutely know them. Okay. So first thing that happened here, around 1920, so this is about 105 years ago. That's not too long ago, I think. All right. Well, my grandfather was born in 1910. Yeah. Okay, so yeah, I feel old. 1920, okay? This guy, Griffith, okay. So he was studying a strain of bacteria that would cause pneumonia. Okay, pneumonia was always a big problem. Remember, this time they didn't even have antibiotics around, so people were dying. Okay, so he started to study 2 strains of bacteria. He looked at a smooth strain. That means the bacteria, when he looked at it, the bacteria looked smooth. So here's the DNA. Okay. Bacteria looks smooth. So he looked at the smooth strain under the microscope, and he figured out that this strain would make the mice die. So that would be really fatal. Okay. And then he found other type of bacteria that was rough strain because the bacteria looked a bit rough like that. So here's your circular DNA. So the rough strain, when he infected the mice with rough strain of bacteria, the mice would get a bit sick, but they would survive. It wasn't such a big deal, okay? And then what did he do here? He decided to run an experiment, and he said, if I kill the smooth strain, so the smooth is the bad guy, okay? So S is for smooth and is for bad guy, okay? So he killed the bad guy. How did he kill it? How do you kill certain bacteria? You can boil them. Yeah, he heat them up, okay? So he just put in a solution and put it in a fire because, no microwave back then. So he boiled the bacteria, he killed them. And then he tested the smooth strain. He tested to make sure the bacteria was dead. So he injected some mice and the mice survived, so bacteria was dead. So what did he do? He also infected the mice with the rough strain, and then something happened. There was some transformation in the bacteria. The good bacteria became bad. Okay? So here's the experiment. Okay? Here's the experiment that he did. Okay? So let's see here. This first guy here, that is the rough strain. Remember, the rough strain is the good guy. He injected the mouse. He waited for a few days. The mouse lived. So no problem. So here is another experiment. He got the S, the bad S, right, strain, injected the mouse died. Then he killed the S, okay, so that's a live S. Okay. He killed the S, he injected the mouse, and the mouse lived. Okay, happy, okay. And this is the part that was really, very important. He did the experiment. He got the dead S with live R. R is the good guy, right? He injected dead S with live R, and he ended up getting live S. What happened to the mouse? Okay, how many people think, I put this crude here because everything, you watch the crudes? Yeah, everything is like you try everything and you die, right? Okay, so that's why I like this guy. Okay. So, how many people think the mouse lives? Okay, how many people think the mouse dies? Okay, how many people don't think? Don't answer that. Okay, so the mouse dies. So now this guy Griffith is like, something is happening. The dead S somehow passed some kind of information to the live R, and live R became a bad S. Okay? Live R became bad. And believe me, it's always like this. It's always a good one that becomes bad. Don't ask me why, right? The good one, the bad one becoming good, I don't know. So, that's your bad S. The dead ass came back to life. All right. So the question here is, what is the transforming factor that was recovered from the dead ass to influence the phenotype of the good R? Okay. So he didn't know at that time. Then later came these other two guys. So this is later. This is 1944. That's actually the year when my mother was born. Okay, 1944, okay. So these other three guys, they really figured out that DNA was the genetic material for the bacteria. Okay. So how did he do that? So this is from your book, right? So what did he do that? He got the S strain, and he started to separate different parts of the bacteria. So in one tube, he got only RNA. In another tube, he got only protein. And then in the third tube, he got only DNA. And then something else makes up bacteria. Is a very simple life form, right? You either have carbohydrates or you have lipids. So in one tube, he took the lipids, and in another tube, he got all the carbohydrate. Okay? So what happened? He separated all the parts that he thought they were bad parts from the X strain. Then he added 2 R, live R, good bacteria. And then he wanted to know which one of these pieces could make good R become bad X. That's all they did. Okay? So now let's see here in more details what he did. Okay? So here you have heat-killed S strain that got the pieces solubilized. So over here, you're going to have DNA, RNA, lipids, and carbohydrates. Okay? Okay, this stage was actually saying that he got rid of lipids and carbohydrates. They figure out lipids and carbohydrates were not going to do anything. So then what happened here? In one test tube, he took a portion of this and he added proteinases. What are proteinases going to do? Proteinases, they are enzymes that break down proteins. All right. So in this tube, is it going to have any protein? No. So what is left in the tube? Nucleic acid. Well, he took out lipids and carbs, so they got rid of those already. So here you're going to have nucleic acids. What kind of nucleic acids? DNA and RNA. Everything in there. So you just got rid of proteins. Okay. In another tube, he added ribonucleases. So ribonucleases are going to get rid of ribonucleic acids, gets rid of the RNA. So in this tube, what you're going to have? Protein and DNA. All right. And in the third tube, he added deoxyribonuclease. He got rid of DNA. So in this tube, he's going to have RNA and protein. All right. So now he added those things to Erlenmeyer flasks that had R strain, live R good bacteria. Okay? So he added this here, another tube he added this there, another one he added this over here. And he found out that the 2 cultures that he added that contained DNA, those 2 cultures transformed the bacteria. But the culture that had killed the DNA was not capable of transforming the bacteria. So he figured that they figured out that DNA must be the transforming factor. But again, this is an indirect way of figuring out that DNA is a transforming factor. Right? They damaged the DNA, they broke down DNA, and they said, without the DNA, you don't get transformed bacteria. So DNA must be it. Okay? Simple? Cool. Oh, I had the dead S here. Okay. And then you get live S and live S. Okay. All right, so they figure out DNA is the transforming factor. So looking more here at the DNA. What is making that molecule? What is the structure of the molecule? You have a backbone of the DNA that is made up of the oxyribose sugar molecule. So here is your deoxyribose. Remember, deoxyribose is missing that little foot, right? Each one of those sugar molecules is bound via a phosphodiester bond, okay, to the other deoxyribose. So here is your phosphodiester because it has oxygen here. This is one ester, that's another ester. So that's your phosphodiester. Everybody remember organic chemistry? Hopefully, a little bit. So that's your phosphodiester. Just in case you don't remember about pHs, acidic and basic stuff, because it was in your test, right? It was in your exam. Remember the question that said in a neutral pH in water, pH 7, what is the proportion of hydroxide and hydrogen ions? A lot of people got that question incorrect. So just put a little review here for you to go back to your very basic inorganic chemistry principles to figure out what acidic pH means, or what basic pH means, what happens to water, okay? So this is telling you ribonucleic acid is an acid. Remember, acids are hydrogen donors, right? So if you look here at your phosphate group that is close to your 5' carbon, you'll see that this phosphate group has 2 negative charges. That means the hydrogens from there, they got donated to the water environment, right? So this whole thing here is an acid. And the other phosphate groups, those have 1 negative charge each, okay? So this whole thing is an acid because donated hydrogen to water. And acid means that you donate the hydrogen. And pH of acids are what kind of pH? Lower than 7, right? pH, definition of pH is negative log of your concentration of hydrogen. Okay? So here, okay, DNA is an acid. So at pH 7, what is happening here? Okay, the hydrogen from that OH group got donated somewhere else. It got pulled off by a water molecule that was close by. Okay? All right, so here is a review of what that actually means when you donate hydrogen. So remember, pH is negative log of hydrogen ions. When you look at dissociation of water, you have this. Right? Remember this dissociation of water naturally happens. And then you have a little constant over here, constant of water dissociation. That constant is called Q equilibrium. That's your equilibrium constant. How do you calculate it? Products over reactants, right? Simple chemistry. So your equilibrium constant is going to give you a number, okay? Now to make things simple, we get the concentration of water, because that's basically not changing, and you plug it into this equilibrium constant, and you end up with a constant of water, with a constant of dissociation of water. That constant is equal to 1 times 10 to the negative 14, okay? When you have pH 7, it means that both of those concentrations are the same. Because if you algebraically solve for each one of those 2 concentrations, you're going to get 1 times 10 to the minus 7. Okay? So at pH 7, both of your hydroxide and hydrogen ions are going to be at the same concentration. So here is your pH 7. So that was the answer to your question. Okay? When you go to acidic side, you're going to start to increase your amount of hydrogen, right, of your hydrogen ions. So if you go, if you have a pH 2, that is like quite acidic, okay? Concentration of hydroxide is going to be a lot lower, but both of them, remember how exponents get factored? You're going to add the exponents, you're going to end up with 10 to the minus 14 all the time, because that's your constant of dissociation of water. Was that a quick refreshment? Yeah, everybody's seen this before, right? Yeah? This is when you put it together, basic chemistry, a bit of organic and all the other stuff. That's the fun class. So here, at pH 7, you have the dissociation of those hydrogens. Each phosphate group in the middle of that DNA has 1 negative charge. Each phosphate group at the 5' end, which is right there, this phosphate is right there, has two negative charges because one of them is not connected to the next nucleotide. Okay? And what happens here at the 3' end? What's going on at the 3' end? This is the 5' end. Because remember, here is your one strand of DNA. This is your backbone, right? Phosphodiester bond. So one end is going to be at your 5' carbon. We call it 5' because the carbon without the prime is part of your base ring. Okay, so this is carbon 1, carbon 2, whatever the number is. So these guys are the prime ones, the ones with the sugar. So here is the one that is a 5'. You look at the other side of your backbone, and here is going to be the one at the 3'. See, 3' here should be connected to a phosphodiester bond, but this guy here at the end is not. So what is connected over there? The hydrogen. There's nothing in there. It's just a OH group over there hanging. Okay? And this actually should be making sense because when you look at DNA polymerization, we see that all DNA polymerases, as well as all RNA polymerases, are going to add groups 5' to 3'. What does that mean? It means that a group is going to come in here and it's going to connect to the 3' side. Well, that makes sense because when you're adding things to this side here, you don't have to deal with bulky phosphate. The thing is ready to have a living group, which is your hydrogen over there. It's an easy reaction. Okay? So the structure here of your backbone should already be telling you that you can add things to your 3' when you're polymerizing this DNA, but it's kind of difficult to add things to the 5'. You need to use tricks to add things to your 5'. Okay? All right, so 2 negative charges at your 5', 1 negative charge in the backbone. All right. So here, one of your 4 bases gets attached to your sugar molecule. Okay? And if you know which ones are the bases that go into DNA, we have that before. What about the bases, right? They are cyclic molecules and they have nitrogen and they have ring structures, okay? They are capable of donating electrons to other molecules, okay? That's why they call them bases, okay? They are not soluble in water. So if they are not soluble in water, they are going to be pushed away from water. Okay? So they're going to be more to the inside of the molecule. Insoluble in water means that they like to interact with each other, but not with water molecules. Okay? Some of them have 1 ring, some other ones have 2 rings, and they're going to form those nucleotides. Now, so here is your 5 carbon sugar, and you know it's deoxyribose. You got to know the 2 rings that the ones that have 2 rings, those are your purines. The ones that have 1 ring, those are your pyrimidines. I like to think that the guys here with 1 ring, they are shorter than the guys with 2 rings, of course. Right? So these guys here that have only 1 ring, they want to compensate because they are smaller. So they kind of have a bit of a Napoleon complex. And since they are smaller, they at least end up with a big name. So the guy with that is 1 ring ends up being a pyrimidine. Okay. So the guy that is taller, you kind of have a better sense of self. It's like, okay, I'll just be the purine. So small guy, pyrimidine. 1 ring, yeah. Oh, okay. So here, there is a bit of fun stuff to the nomenclature. I'm not going to test you on nomenclature. I just test you on the basics stuff. So if you want to read here, you know, if you have a nucleoside that does not have a phosphate, you have a nomenclature that ends up like adenosine. And if you add at least 1 phosphate, you end up with a nucleotide. Right, so this is important. At least 1 phosphate, you have a nucleotide. So here, Okay. Okay. Also, you know that only one phosphate is going to be part of the backbone of your DNA, as well as backbone of RNA. So one phosphate on your nucleotides. Okay. So things that get incorporated are monophosphates. One phosphate that gets into the backbone. In case of DNA, you have your deoxyribonucleoside monophosphate. This thing has weird names. Okay, so here, more on nomenclature, okay, blah, 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 you can read that on your own. All right, so here are your deoxyribonucleoside monophosphates that get incorporated into the DNA. Okay? Okay, all right, so this is important. Why is it that ends up only with one phosphate in the backbone? Well, because you need a phosphodiester. If you put a bunch of phosphate groups, you're going to have a lot of electric hindrance, and it's not going to be in the right shape. And it takes a lot of energy to get these groups together, to synthesize the nucleotide, to synthesize the nucleic acid. Okay? How much energy it takes? It takes actually the energy of 2 phosphates. So when your nucleotide is getting close to be incorporated in your nucleic acid chain, it comes as a 3-phosphate. Okay, so here's phosphate, it's phosphate. So 2 of those phosphates get cleaved, so the energy of these 2 phosphates are used. The energy is used to incorporate your nucleotide into the DNA chain. Okay, so 2 phosphates are used. We call that pyrophosphate. 2 phosphates together is pyrophosphate. Okay. Oh, so here is the one that you should know which one is ribose, which one is deoxyribose, right? So just in case, you remember with the little guy that has 2 feet and the little guy that has 1 foot, right? So here you have, the 5' carbon is the arm, the C1 prime is your OH group, and your ribose walks smoothly, and your deoxyribose has a little limp because it's missing a foot. Deoxyribose, not deoxyribose. I did put a link over there. I forgot to check the link. Most likely it works. So here, again, DNA structure. Again, here you see your pyrophosphate is getting cleaved. Energy of 2 phosphates is important to bind here from your 5' right here to 3'. Always F to T. Okay, just think about the alphabet. F comes before T in the alphabet. F to T, that's how you do polymerization. All polymerases will do that. Okay. So here, 5' to 3'. Okay, we mentioned that. But I have a little movie here. All right, so more on the backbone. The inside of the backbone, remember the outside you have all these bulky negative charge phosphates on both sides, okay? Remember those nitrogenous bases, they are not soluble in water. So they're going to be away from the water and they like to interact with each other. So they're going to be facing the inside of the molecule and they're going to interact with each other. Okay? Now, this interaction is really important because they cannot really have a lot of distance between the 2 backbones and they cannot be really, really stuck together. So the interaction is going to be alternating between nitrogenous bases that have 2 rings and 1 ring. So 1 ring with 2 rings, 2 rings with 1 ring, 2 rings with 1 ring. You're always going to have a total here of 3 rings that are interacting in the middle. Okay? And this is important for later when you look at DNA repair. Okay? So here, the bases are facing the inside. They have hydrogen bonds that are going to interact between the 2 bases. Now, let's pay attention here. What is important for these hydrogen bonds? Okay. Oh, another thing also, the direction, the orientation of the backbone One backbone is going to have the 5' end that is going to be at one side, and the other backbone is going to have the 5' end that is going to be on the other side of the molecule. Okay? And the main reason for that is that remember this guy has two negative charges. So if you have another 5' end over here, two negative charges are going to be repelling each other. That wouldn't work very well. Okay? So bulky negative charge one side, bulky negative charge on the other side of the DNA molecule. Okay? So that is your anti-parallel conformation. Bases are going to lay flat right in the middle. Then, oh, another question. The base pairing in DNA is stabilized by what? Okay, make sure that if people are giving me the wrong answer, consult your friends and get the right answer. This is the time when you can bank those extra credits. Anybody need more time? No? Okay, what's your answer? B, that's your hydrogen bonding. Okay. So here more when your hydrogen bond story. Okay. So let's look here. When you have here, remember this guy is the shorter one, with the taller one. So this guy is the purine, that guy is the pyrimidine. A bind to T, C bind to G. I'm sure everybody heard that. And the question is why? Okay, so now it's not just memorizing A binds to T, C binds to G. That's brainless. You got to think why. Okay, so what happens here? Look here at the structure of adenine. You're going to have here nitrogen that has electron clouds. So this guy is electron negative. Okay, and then he's interacting with a hydrogen that is bound to another electronegative nitrogen. So you need this hydrogen to be bound to electronegative element in order for it to be part of this hydrogen bond. Okay, and then you say, wait a minute, look at this other guy over here. This is electronegative nitrogen. It has a hydrogen. And this hydrogen is interacting with this other electronegative oxygen right there. So you have those hydrogen bonds because you have two electronegative atoms that want to interact with the hydrogen. And then you look down here and you say, look, there's a hydrogen right there. This is an oxygen right there. Why is it that you don't have a hydrogen bond that goes this way? Why no hydrogen bond down there? Huh? Here, you see down here? Can you see back here, down here, all the way at the bottom? Okay, so this part over here, right there at the bottom, right here. Okay, sorry for you guys. Oh, you know what? I think I can just do this. Okay. All right, so these guys right here at the bottom, you see a hydrogen right here? I should make an emphasis on this guy. Everybody sees a hydrogen there? Yeah. You see oxygen right there? Didn't I tell you need electronegative oxygen to make hydrogen bond? Why you don't have a hydrogen bond down there? Because this guy here is bound to a carbon. Carbon is not electronegative enough to make this thing to become a hydrogen bond. Okay? So when you look at A and T hydrogen bonds, you're going to find only two of them. Okay? Now this principle is important when we get to mutations. You're going to see a few things that can go wrong And believe me, a lot of stuff can go wrong. It's really amazing that we're not more messed up than we actually are right now. That was supposed to be an optimistic statement. Okay, so let's see what happens with the CG story. Okay, so here you have guanine, here you have cytosine. Okay, look at this. NH hydrogen bond, NH hydrogen bond, and then, oh, look, this guy here has oxygen. Now we have nitrogen, oxygen. You can have 3 hydrogen bonds between your C and your G. If you have 3 hydrogen bonds that are having this electrostatic interaction, I want 3 bonds. There we go. Electrostatic interaction. I got confused for some too many fingers. If you want to pull them apart, it's going to be harder. In comparison to only 2, that's easier. 2 hydrogen bonds, you can pull that electrostatic interaction apart a little bit easier. Keep that in the back of your head, because when you look at gene expression, you're going to look at promoter regions in the DNA, and you're going to find that a lot of the promoter regions have a lot of A and T interaction, because you can pull them apart a lot easier. Okay? So that's the story with the hydrogen bond. All right. So here is also, oh, I had it emphasized. See, I forgot about it. So here we go. Your hydrogen bond is not doing the bond over here because this guy is carbon. Yeah, that's what happens when you don't review your slides just before the class. All right. So, okay, is there, everybody understands that, right? All right. So here we go with the base pairing. So there is more about the structure of DNA. So here we see the B DNA structure, and it is a right-handed helix, is a double helix. Everybody knows that, right? And the way that this helix will wrap around itself, you're going to end up with a piece that has a bigger indentation and another little piece that has a smaller indentation, and those are going to alternate. Big, small, big, small. So you call the big one the major groove, you call the small one the minor groove. Again, remember that the strands themselves are oriented in anti-parallel way. Okay. So the bases in the DNA, they're going to do the hydrogen bonding with each other, but they're not only interacting with each other. Remember, this is aqueous environment. They are also going to interact with water molecules that are all around. And this is also interesting because, remember, you have here a lot of electron cloud. So the orientation with the interaction of water, you're going to have hydrogen bonds. Okay? You're going to have electron clouds here. You're going to have positive charges coming this way. And you're going to have a bit of a charge separation. Remember that easy water, the fourth state of water? This is actually what is happening that keeps the DNA molecule together. Okay, so when you look at the inside of the nucleus, you find this gel state. That's actually the easy water. Okay, you do have the charge separation. And remember, after the backbone of the phosphate, you're going to have this negative charge that's going to be surrounded by positive charge. Okay? Okay. So here is an interesting paper that came out not too long ago. So this is 2020, showing interaction of bases with water molecules. So you might think that this information is like there forever, but it's actually not. So interaction of DNA with water molecules, these scientists just came up with this paper in 2020. This is only 5 years ago, right? So they're still finding out a lot about water, believe it or not, right? Another question. Hey, I tell you, party. Hey, it's Friday. Okay. Which of the following correctly describes the base pairing in DNA? I'll actually put up a little video of this guy doing the wrap of the DNA. It's very cool. Have you seen the DNA wrap? No, not wrap, I mean accent. C's, 1, 2, G's, and G, I didn't. Okay. All right, does anybody need more time? No, everybody gave me the correct answer. 7 people are giving me wrong answer. Change your minds. Okay, what's your answer? C. Okay. All right, so here, a little bit more about DNA structure. I mentioned the middle one is your B form. That's the most common form of DNA. Remember who discovered the DNA? The shape of the DNA? That was Watson and Crick with data that they stole from Rosalind Franklin. Yeah, you know that story? No? Oh, it's interesting. Yeah, they stole the data. And amazingly, in the same Nature paper that this thing was published in 1953, Rosalind Franklin managed to publish a paper showing the A form of DNA in the same paper, with, of course, her own data. Of course, the rest of the data had been stolen. And then who got the Nobel Prize for discovery of DNA shape and molecule? Watson and Crick. Why was it that Rosalind Franklin did not get also the Nobel Prize? She was dead. And they say that people cannot get the Nobel Prize. Anyway, don't say. Is this thing of data stealing a thing of the past, and human beings now are much more evolved that this kind of thing never happens again? We wish. Let's not get into that story. So, so what happens here? bDNA. That is the part that is hydrated. Is a lot of the DNA that you find in the cells is actually bDNA. Now, interestingly, when you dehydrate some of that DNA, you end up with the A form. This is why Rosalind Franklin had published the A form, because the samples of DNA that she was using to do the X-ray crystallography, they were very much dry samples. So she ended up with a very much desiccated form of DNA. So she ended up seeing the A form of DNA. This, I have here that you find this A form in spores of bacteria, okay? And the difference between the A form and B form, of course, is dehydrated. The groups get smaller. You don't have a lot of space for other molecules to come and interact with the DNA molecule. Okay, so it gets shorter, it gets like shrunk into this smaller, compacted version. Now, what we know in recent studies is that you also find A form of DNA in our own cells. It's not something that is exclusive of spores of bacteria that are dormant. You do find A form of DNA in our own cells. And one major reason for it is that if all of your DNA in the nucleus is B form, you're not going to have enough space in the nucleus to pack all the DNA that you actually have there. Okay, so areas of the DNA that are not being used for certain cell types, these areas get packed into A form. Okay. Besides that, you also have this weird Z type of form of DNA, and this happens to be a left-handed helix instead of right-handed helix. So this is your right hand, and this is your left hand, kind of like that. Then you try to walk with it. Okay. So left hand kind of looks inverted. So you have different conformations for this type of packing. Okay. And you also find Z form of DNA inside eukaryotic cells. And interestingly, you find a lot of Z DNA form close to genes that are supposed to get expressed. So before you find the genes, you find some cDNA, which is like a flag that is going to tell all the proteins in the cell that a gene for expression is close to this weird form of DNA packing. Okay? So you do have the 3 forms of DNA in our own cells, but the B form is the form that is most common when you look at a gene expression. Other thing that is different between zDNA and A and B DNA is that with the zDNA, you find a lot of CG base pair. And by CG, lots of hydrogen bonds. So it's going to be really tough to pry those individual strands apart. They like to be glued together. So here's a bit more of a spill on where you find the DNA. A form you mainly find in spores of bacteria, is a more compacted form, and is important for those bacteria to be in this form of DNA compaction, because they are at its dormant state. They're not really active, and usually they're out exposed to the elements, meaning that they could be exposed to sunlight, UV light, right? So if the DNA is not very much compacted, they could get DNA damage because of UV light. So you find this form most commonly in spores of bacteria. Okay. Right. So what is important here? All right. So I mentioned Z form of DNA. Okay, so this part is actually, I have to review this, okay? We know that zDNA exists because there are proteins that bind zDNA, okay? So this slide actually I had prepared before we found out about the presence of this zDNA in cell. So this slide is telling you most likely zDNA is present in the cells because there are proteins that actually interact with this form of DNA. So now we know that zDNA actually is present in cells, especially before genes that get expressed. All right, so DNA contents in prokaryotes. You have 100% of a genome that is going to give you gene expression that is dependent on 3 forms of RNA. Now, when you get eukaryotes, you're going to get gene expression that is based also in 3 forms of RNA, but you have a lot more RNA that is part of the story, okay? So when you compare to 100% of genes in prokaryotes that make use of the expression of their genes with this type of RNA, when you look at eukaryotes, you're going to have only about 5% of the genome that is going to be part of these coding regions. Okay, so 5% of the eukaryote genome is going to be expressed as genes or protein encoding genes. The other types are doing something else. Okay, you have about 15% that is non-coding regulatory regions of the DNA, so non-coding regions. You have other parts that are non-coding, non-regulatory regions. And you get more of that. 1 second. Yeah, so the no coding, I didn't put in the specific names for those RNAs, but you can imagine only 5% is making proteins, so everything else, 95% is involved in something else. It's not there just because it's there. It's useful, but it's something else. Okay, also, we know that eukaryotic DNA is inside of the nucleus. So you have a nucleus membrane, that is a double membrane, that is circulating all the DNA. Okay. Okay. So let me see what is important here. All right, so we know that we have the double helix. All right, so one thing that you also, of course, have to remember here is that your prokaryotic DNA is circular and your eukaryotic DNA is linear. Okay. When you look at the prokaryotic DNA, you don't really have it as a chromosome structure, but you do have association with other proteins. You just don't have a chromosome type of structure. So here, let me see. When you look at eukaryotic genes, you can have eukaryotic cells that have a very small number of chromosomes, meaning like if you look at ant DNA, you're going to have only like 1 or 2 chromosomes. If you look at other species like butterfly, look at that, 450 different chromosomes in a butterfly. That is a lot. How many do we have? 46, 23 pairs. Okay, so butterflies like 450. That's amazing. So eukaryotic DNA is packed into these compaction structures. They are the nucleosomes. And we're going to look at them more. Okay? So here, see, okay, I'll take 3 more minutes here. All right, so when you look here at your prokaryote, you have your circular DNA, and you don't have those membrane-closed organelles, but you do have a place in the bacterium that is where the circular DNA likes to hang around, okay? So you call that region nucleoid region. When you look here at your eukaryotic DNA, is packed linearly and it is inside of the nucleus. When you look at the protein also, as we're going to see with the eukaryotes, you're going to have a lot of association with specific proteins. So here is this part of the chapter that you're going to see how DNA is packed and implications of the linearity. Okay, so one piece of chromosome during interphase. Remember that when you start to undergo cell division, first thing that you're going to do is you're going to replicate your DNA, right? So when you replicate the DNA, you're going to have still this DNA that is replicated but connected. So each one of these is going to be a sister chromatid, okay? So when you're undergoing cell division and you have duplicated DNA, one piece here you call these chromosomes. Same thing, like one piece here you call this a chromosome. So the difference between interphase and synthesis or cell division is that this piece of chromosome is going to have 2 sister chromatids. Okay, so it's going to be condensed and ready for cell division. All right. So here again we have 23 pairs of chromosomes. The numbering of your chromosomes start with the biggest chromosome. So the biggest one is number 1, and then it goes smaller, 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 and all the way to chromosome 22. That's the tiny one. And then you have your sex chromosomes. Then you're going to have either XX or XY, right? Sex chromosomes are actually bigger than the smallest chromosome. Okay. You inherit 2 chromosomes, right? You inherit one from your mom and one from your dad. So we are diploid organisms, okay? So the chromosome that you inherit, one from mom and one from dad, they could have genes that are slightly different from each other. So you could have slightly different alleles present in one or the other chromosome. Sometimes they're the same, sometimes they can slightly different arrangements. All right, so we're going to stop here at the chromosome territories because I see people starting to pet and running away. And we're going to pick up from here.